Hi everyone, welcome to part 2 of the Bioinformatics COMP0082 module. This is going to be quite a short second part of this module. It's going to be sort of the last 2 weeks of term. We're going to cover high throughput techniques, biological networks, biological systems, and how you can actually apply machine learning to these to understand, for instance, particular diseases. So that's the sort of overview. I should introduce myself. I'm Kevin Bryson. This is my email address if you want to email me. If you're watching this video, you've probably noticed all the teaching material is on Moodle. We're going to have a Q&A session at 12 noon on Wednesday, so you're familiar with that. I think it would be a good idea as you watch these videos, if there's anything that you don't actually understand or it's confusing, it would be good to take a note of it so that you can ask it at the Q&A session. I think the Q&A will use Zoom and people can ask questions and largely anonymously, because you can post it just to me, the question, and I can read it out and answer. So, or you can, if you want, appear in the video and present it in person. So we'll see how that goes. So what is this part of the course going to be about? Well, with David, up to now, you've been looking at individual biological parts, DNA, proteins, etc. And he's explained how machine learning can be applied to these biological components to make predictions about them. For instance, in the last few lectures, he's explained how CyPred works, which takes the amino acid sequence of a protein and can predict its secondary structure. It can predict which parts of the protein go into alpha helical structures, beta sheets, and coil structures. Now, this is very valuable for the bioscience community. For instance, if we were looking at sickle cell anemia, then you may be aware that that's caused by a single mutation in the hemoglobin protein, which causes it, once it folds into a structure, it causes it to have a sticky hydrophobic patch on the surface, and then the hemoglobins all stick together, causing the red blood cells to become sickly in shape instead of the concave sort of ball shapes. And this causes a number of other problems. So if we didn't actually know the structure of hemoglobin and we had limited information just about the sequences in terms of the mutation, we could actually use something like AlphaFold to actually predict the structure of the hemoglobin, see where the mutation was on that structure, and then hypothesize it's on the surface. It might cause it to stick to other proteins of itself, leading to disease. So these are valuable techniques. But a lot of diseases require understanding of more than a single gene or protein. In fact, I think most diseases, cardiovascular, cancer, they're complex diseases. So this is really what we're going to start moving towards. What do we need to know to actually understand these complex diseases, and how do you start applying machine learning to these complex diseases? So to give you an overview of the part 2, this lecture is very much just a motivational lecture, telling you about biological systems made up of biological networks and the techniques, the data, experimental techniques which can give you information about complete biological systems, often called omics high throughput technologies. So we're just going to really give you a bit like David gave you the context of biology in the FAST lectures before going on to machine learning. This is very much giving you the context of the biology involved in understanding biological systems, a sort of systemic understanding. And then we're going to, in lecture 2, move on to more detailed description of a single omics technology, probably the most important one, called transcriptomics. So that's how much the genes are being transcribed. From all of this information, one of the things which will become apparent is that the data for biological systems is complex. So we'll then in lecture 3 look at data representation. How do we represent the semantically rich omics data? How do we represent its biological experiments, biological networks? And you see this is quite a complex topic, but we need to to do sort of proper machine learning of this type of area, biological systems. We need to have a better representation, understanding of data than just a big data table full of floating point numbers, the usual sort of understanding that machine learning has of data. We need to have context to the data as well, but we'll see that. And then to finish off, lecture 4 is going to look at essentially how all of the different techniques for applying machine learning to omics data. This is still a field which is pushing forward. We're going to use simple machine learning techniques. I'm hoping the other modules will be teaching you sort of the more advanced machine learning, but really just to provide that interface of how to apply machine learning to actually omics biological data. So some motivation for why we want to sort of understand these systems. There's quite a lot of interest at the moment in actually determining personal genomes. You can basically pay £40 and get companies like 23andMe to actually give you back your personal genome. And you can find out things like what isoform you have of the ApoE gene. So ApoE stands for apolipoprotein E, and it's essentially a protein which is involved in carrying lipid, fat around the blood. And there are 3 major isoforms, 2, 3 and 4, and they all have different sequences of the gene. So they result in proteins which have slightly different amino acids. And for some reason, these different isoforms, after going through the complex system which is the human body for 60, 70, 80 years of processing, can lead to an increased risk of Alzheimer's disease. So in particular, ApoE form 4 increases the risk of Alzheimer's disease, and ApoE2 decreases the risk. How that happens, nobody really understands. It doesn't, even though it is one gene involved in this predisposition to Alzheimer's, it's certainly not a single gene involved in Alzheimer's disease. It's a complex network organization of lots of parts leading to this. Another example, well, so this is healthy skin, and we all know that if you get sun exposure in particular, this can change the genetics of particular DNA in the skin cells, which can then lead to melanoma, skin cancer. How this happens, again, is a complex mechanism. There are oncogenes which cause cancer when they're mutated, and there are also tumor suppressor genes which suppress cancer normally, but when they're mutated and don't function anymore, they can also cause increased likelihood of cancer. But this is also involving lots of other processes. So this is the area we're in. We're in the area of sort of understanding complex diseases. We're going to start off with, so human cells, which both Alzheimer's and skin cancer involve, are incredibly complex, and I'm hoping to show you just how complex in this lecture. But I think it's good to start off with a simpler example. Again, it's disease related, Streptococcus pneumonia. So this is a micrograph, a coloured micrograph of these bacteria, and they're carrying these little chains. And they were first isolated by Louis Pasteur, but also the US Army physician George Sternberg, 1881, due to the role in causing pneumonia. These bacteria actually are part of the normal respiratory flora. We all have these bacteria at the moment, and they normally live quite happily in our nasopharynx, and only occasionally do they become pathogenic and cause disease. So they do cause 1.6 million deaths annually, mostly in people who are immunocompromised, have weaker immune systems, under fives and elderly. How does that happen? Well, it's using this, so this represents the bacterial sort of membrane around the bacteria. So this is the extracellular space outside the bacteria. This is the intracellular inside the bacterial cells. And these are representing the DNA within the bacteria. So the DNA is a single long DNA molecule, but this is showing you individual genes. So we've got a COMP-A and B gene, and then COMP-C, D, E, and COMP-X. So these are DNA sequences. What the COMP-C gene is producing is this CSP protein, and this stands for competent stimulating protein. And this is actually got it's exported outside the cell, and it's a small peptide. It's short 20 amino acid peptide, and it actually binds onto and activates this COMD transmembrane protein receptor. So when it binds to this, it causes the phosphorylation of another protein called COME. So these are all coming from these genes. This is E, this is A and B going to this transporter of this peptide, and COME is here. This then phosphorylates, adds a phosphate to one of the amino acids in COME, and this then acts as a transcription factor, which actually activates all of these genes again. So that the system actually is got positive feedback. Once there's a certain amount of CSP, it binds onto here, activates this chain, this signaling pathway, which activates all of the upregulation of these genes, which makes more of this, more of this, more of this. So it then turns the whole system on suddenly. How is this? It seems a strange mechanism that it just sort of cycles with itself, but actually how the bacteria uses this is to sense how many bacteria are currently in that part of the body, the nasal pharynx, for instance. As there are higher density of bacteria, you get a higher density of CSP, and all of them get turned on in terms of the quorum sensing mechanism, this is called. So it senses the level of bacteria within that particular niche, the nasal pharynx. And this is actually what then drives it. If the bacteria are not in control by the human, and it increases to a certain level, it can then become pathogenic. It knows that it can then sort of become invasive and pathogenic. This turning on of the COMX gene is actually what causes it to become pathogenic. It makes it then invade the system. Knowing this overall system, one of the things we can do is design peptides that stop the process. So we can design a peptide that looks like CSP, that binds to the COMD, but it doesn't allow it to turn on the signaling pathway mechanism. So our drug peptide uses up binds very tightly to COMD and stops the natural CSP binding. So it doesn't allow the streptococcus actually to become pathogenic. So this is the sort of example system of a disease system which we're trying to understand using these network biology and omics techniques. So this actually highlights some of the aspects in David's part of the course. You've hopefully looked at protein sequences and structures, and also genomic DNA sequences. My part's going to be about gene expression and also the biological networks. So one of the key omics technologies we will learn about is transcriptomics. This measures the activity of different genes in the organism. Essentially, it measures how much messenger RNA is being produced from each gene. So in this particular example, we've got the COMC gene, which is here, this tiny little one, and it's transcribed into this messenger RNA for the COMC gene, which is then translated into the CSP protein itself, the competent stimulating peptide. So we can actually, using transcriptomics technology, measure how much expression there is of the COMC gene, and similarly how much expression there is of COM-A and COM-B, so how much of this exporter is being produced, or how much COM-E is being produced, how much of this protein. So it's actually measuring the expression level of all of the genes in the organism. So this allows us to do experiments. A particular experiment they have done in the paper that I'm going to cite is that they add synthetic CSP peptide to a sample of Streptococcus pneumonia, and then they measure how the expression of the genes changes over time as a time series. Proteins called transcription factors, like the COM-E, bind onto particular DNA sequences upstream of genes to turn them off and on. So you can see that COME, once it's phosphorylated by an activated signaling pathway, it binds onto here, here and here and turns on all of these genes. And if we do that experiment, we get this data. So this is Streptococcus pneumonia, where they've added in CSP, synthetic CSP at the start, and then measured it every 2.5 minutes up to the time 20 minutes. And it's in this paper here, identification of competence pheromone response genes in Streptococcus pneumonia by using DNA microarrays. And what you actually discover is that there are some genes, like these, which basically don't change very much over the time series, so the black line here. And then there are genes which actually get activated. This yellow colour is showing activation of the genes, increase, very early, after 2.5 minutes. And then they actually turn off again. So this is the red line here. They go on, and then they turn off again, the gene. And then there are other genes which get activated a bit later, about 10 minutes, and then they go on and then off again. So this is, well, there's the green, which has a slightly different profile, but then the blue, it gets turned on later, and it gets turned off again. These actually get the names early genes and late genes in this process. So how does this omics data, this transcriptomics data, relates to the biological network? Well, what the experiment has done is that it's added CSP. This will have activated the COMD receptor on all of the bacteria, and this would have then phosphorylated the COME protein and activated the signaling pathway, turning on all of these genes. And these turn out to be the early genes. The ones that are turned on early, 2.5 minutes, are these ones. And it indicates that from adding the CSP, it takes about 2.5 minutes to turn on all these genes. What are these genes then? Well, once COMX has been activated, its expression, it then goes on and turns on sort of 10s, 20, well, 50, this paper shows actually 100 different genes downstream from it. This just shows some of them. So this is actually essentially another 2.5 minutes after that one. Well, it's a bit faster maybe. So it's about maybe 3 or 4 minutes to turn on a gene fully, and then this is about 10-ish minutes. So 4 minutes turns on this gene, and then 4 minutes later the expression level of this is increased and the protein's been synthesized, and it turns on all of these other genes, and this shows these other genes going on. So you can actually understand the biological system in terms of the biological networks and the omics data. We can go a bit faster and actually look at the genomic sequences. So this shows ensemble bacteria, which is a genome viewer, and it shows the Streptococcus pneumonia. You possibly don't see it because it's quite small, but this shows the COMC gene in the genome. And we can look at all of the upstream DNA of these genes and use something called a motif detection tool. So that essentially uses machine learning. It looks for patterns of DNA sequence which are common between all these genes, and it detects this pattern. So there's this sort of GAG pattern, T, over this sequencing, and then there's this strong AA, TG, T, and then TATA pattern, TA, TA. And this is the pattern that is basically bound by the phosphorylated COMP protein. So all of these genes have this pattern at the start of them, and the phosphorylated COMP essentially binds onto this pattern in the DNA, this pattern of nucleotides, and then activates all of these genes. So you can sort of see how the biological networks, the gene expression data, and the genomics sequences, in particular the control sequences, the cis-regulatory control sequences, actually all relates together to give this biological system its mechanism for working. I think that's probably a good stage to pause these videos, which I'm going to try to keep shorter. But I think we've covered a lot of details in quite a high level, and there's probably quite a number of questions that have arisen from this. So I think it would be good for you to look at this video possibly again and note down questions that arise from it, because you may all have different levels of the biology aspects, and I'm sure some of you have questions arising from this. Okay.